Hi, so we are doing Standing on the Shoulders of Giants as our topic, and we'll begin now. In 1953, Watson and Crick published their characterization for the salt of deoxyribonucleic acid, DNA. The structure they proposed consisted of a double-stranded helix connected by hydrogen bonds. The bases adenylene and thymine are always paired together, and guanine and cytosine are always together. This is an image of Watson and Crick working in the lab, where it can be seen that they created a model of the structure DNA using a technique by Linus Pauling, as they did not perform any experiments but rather put together the work of others like a puzzle.
And this is just a brief timeline of some of the researchers' work that Watson and Crick used when they put their model of DNA together. The first of which is Friedrich Mischer. So in 1869, Mischer was the first person to discover what we now call DNA, but what he initially dubbed nuclein. Mischer's initial goal in the research that led to this discovery was to determine the chemical composition of cells. He used leukocytes from old dressings containing pus for this research. During the process of extracting proteins from the white blood cells, or leukocytes, Mischer discovered a substance unlike any protein or lipid. It was resistant to proteolysis, which is protein digestion, was made up of large quantities of phosphorus and lacked any sulfur. Mischer was able to identify that this substance was contained within the nucleus of cells and therefore named it nuclein. He researched nuclein for many years, even theorizing at one point that it was responsible for the passing down of hereditary traits. However, he later rejected this hypothesis as he did not believe that nuclein was capable of creating enough variety to account for all the variations of life in the world. However, his initial discovery of DNA was crucial for further research to develop many years later. Another researcher responsible for helping Watson and Crick with their model was Phoebus Levene. He was a biochemist and is credited with defining the molecular structure of DNA. In 1919, he proposed his polynucleotide model for nucleic acid structure, and this model was the result of many years' research using hydrolysis to break apart and analyze nucleic acids in yeast cells. The polynucleotide model suggests that nucleic acid is made up of nucleotides that have one phosphate group, one sugar molecule, either ribose in RNA or deoxyribose in DNA, and one of 4 nitrogen-containing bases. These bases are the purines, so adenine and guanine, and the pyrimidines, which is cytosine, thymine, and uracil, although uracil was not identified by Levene. He also proposed the tetranucleotide model, which argued that nucleotides would join together in the same order always, but this model was disregarded as overly simplistic and later proven incorrect. Levene's polynucleotide model was essential for further DNA research to occur and eventually contributed to the work of Crick and Watson.
The next scientist that credited Watson and Crick was Oswald Avery. In 1944, he showed that DNA was a transferring principle and identified that DNA carried hereditary material instead of the belief that protein was the transforming agent. He proved his theory by showing a strong reaction to the diphenylamine test. He also led his team to prove Griffith's theory by completing experiments demonstrating that DNA brought variants of pneumococcus into infectious encapsulated pneumococcus.
In 1950, Chaga published his findings in relation to the base composition of what we know to be DNA. From his experiments, Chaga had 2 main findings, the first being the proportions of adenylene and thymine, cytosine, and thymine. Varying between species, the DNA profiles of each species is unique. Secondly, he found the amount of A equaled the amount of T, and the amount of C equaled the amount of G. His findings proved to be very important for Watson and Crick, as in their structure of DNA, they follow Chargaff's rule.
Rosalind Franklin was the one to discover the double helix strands of DNA, and this was found in 1953. Dr. Rosalind Franklin's DNA diffraction work at King's, her creation of the famous photo 51. This gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical process within cells. This is her finding. This isn't the exact photo, but it does show the double helix of the DNA, which was found by her.
So of the 10 big ideas in science, the one that this topic really centers around is number 9. Genetic information is passed down from one generation of organisms to another. So the study of genetics was founded on Gregor Mendel's plant hybridization, which was a paper that he published in 1865. And it's talking about the Punnett squares and the pea plants breeding and different phenotypes, etc. The discovery of DNA was revolutionary in the field of genetics and opened up a whole realm of new research, molecular biology, epigenetics, etc. The work of Misha, Levine, Avery, Chargaff, Franklin, and Watson and Crick was all essential in the actual development of this big idea. Without their work, we may not even have this big idea to work from. And they revolutionized the field of genetics with their work.
So, out of the 4 big ideas in science, we chose scientific explanations, theories, and models are those that are best fit for the evidence available at a particular time. So, Watson and Crick believed their model was roughly compatible with the experimental data, but rightfully said that it needed further testing. So this shows that their model matched the number 4 big ideas in science, as they believed that it was the best fit for the data at time, but thought it still needed work. However, they believed the works of Pauling and Corrines and Fraser, which was unpublished, model was incorrect, as they dismissed Pauling and Corrines' free chain model because Watson and Crick believed the negatively charged phosphates at the axis would repel each other, and hence they believed the substrate that gives the X-ray diagrams to be a salt and not a free acid. They also thought parts of the van der Waals distance to be too small. And for Fraser's model, they ignored it because they thought it was too vague. They concluded by saying their model was consistent with the relevant data and theory, which is why we chose this theory out of the 4 big ones. So then going on to the next slide, this is everything we talked about from each of our slides. And then in the next one, these are our references for the main ones. And then in the next one, these are our references for the images from each slide. And yeah, we'd just like to say thanks.
Beautiful.
Thank you.
Oh my God.